Now it's class time. I sent out an email last night after deleting a bunch of emails from you guys because you basically crashed my email account by submitting your stuff electronically. So one of the things that COVID last year taught Dr. Judd and I about electronic submission of assignments is that we spend a lot of time with electronic submissions of assignments just managing the files that are coming in rather than spending time actually paying attention to the grading. So managing electronic files, especially when they're not named the way we want them to be named and things like that, it just becomes a huge time suck just to manage the digital files. And so it's easier for us to just get assignments turned in by hand. So we would prefer you to do that. There are going to be a couple of instances where we do ask you to turn things in electronically, but that's mainly so that you can run those things through a plagiarism checker and at least get a first look by a computer algorithm on things like plagiarism and whatnot. We'll talk about those algorithms when we talk about plagiarism, but it just makes our lives much simpler to get them physical. I was looking at the assignments that I did get. So I cleared out my email last night, and then overnight some others came in that were submitted yesterday but didn't make it into my inbox because my inbox crashed. And then I looked at those, and as I was looking through them, they're all named the same file. So I have no idea by just looking at the file name whose is whose. And in two of those cases, they didn't have names of the person who submitted it in the file. So now I have these two papers that don't have a name associated with it. So it's just simpler in many ways and more time efficient from our perspective to just get physical things. The other advantage is when I get your papers, I put them in my satchel. And in order to clear out my satchel, I have to grade them just so I don't walk around with 20 pounds of student papers hanging off my arm. So that also is an incentive. You are standing like you want to say something.
I'm just going to add that it allows us to get through that when you faster. And also, if you're not going to be in class someday and this is in paper, usually you can put that under a door.
Oh yeah, it's totally stuff under our door. If I have to trip over it, I'm likely to pick it up. So that's that. So if you didn't bring a printout copy of your assignment for today, definitely get that to me as soon as possible. There's a schedule change. When we put together the schedule, there have been some changes to the schedule over the last couple of years. And when we looked at it, I had myself down for teaching 21 class sessions, and Dr. Judd had 15. So we've modified things. So the introduction to Galapagos Islands and the intro to natural selection that was supposed to be taught by me is now going to be taught by Dr. Judd, just to kind of even out the workload. I haven't updated the schedule yet, but it will be updated later on today because we just made that change this morning. Questions about anything we've covered so far? Yes, ma'am.
What about the first session?
Yes, good question. So what we do in lab is we have you working in groups on a self-designed research project where your group designs a question, comes up with a question that you want to answer. It's ecological or evolutionary in nature, and then you spend the whole semester kind of developing that question, building a set of experiments or observations, and then you gather the data, you analyze the data, and you present it at the end of the semester. Normally, we would start building the groundwork for you thinking effectively about ecology and evolution yesterday in lab, and we do it by introducing you kind of to ways of thinking about organisms in terms of what organisms need to survive and reproduce. And we go through this fairly long and drawn out process of kind of developing these ideas of what are the components. So for example, we begin with acquiring energy. Every organism needs to acquire energy. And then we say, okay, well, plants and animals acquire energy in different ways. So let's think about plants for a little while. How do plants acquire energy? Well, they do it through photosynthesis. Well, what exactly is photosynthesis? Well, photosynthesis is this equation. It requires carbon dioxide and water. There's also C3 and C4 photosynthesis. There's all this stuff just in exploring photosynthesis. Photosynthesis then also has different implications for water balance in plants depending on whether you do C3 or C4 photosynthesis. Well, one of the other things that organisms have to do is they have to maintain water balance. And so that then ties into photosynthesis. So we build this thing called a concept map that kind of gets at 7 different aspects of an organism's life that it needs to do to survive and reproduce. And we build those out and build this elaborate concept map. We couldn't do that because couldn't really see in the classroom yesterday. So what I'm going to do, I've got you divided up into groups already. What I'm going to do is I'm going to give you that assignment to work on in your group by yourself. If you want me to be there while you do it, I can find a time when we can all meet when I can do that. If not, you'll just do it on your own. I'm going to make a screencast that goes through the acquiring energy part, which is something that we typically do for you to kind of show you how you would think about this. But then after that, in lab, we just cut you loose on the other 6 categories, and you kind of develop it with yourself. It's good to have your textbook with you when you're doing this so that you can draw upon a resource in order to figure that out. But it's nothing that you can't do by yourself once it's introduced to you how to go about it. It's just that we've never had to do it not in person with one another. So one of my tasks this afternoon is to get a screencast put together, get it up on the Moodle site so you can kind of see how you would start, and then you just take it the rest of the way.
Okay.
So yeah, that's just going to have to be done outside of class time. Does that help? Mm-hmm. Yeah. Okay. So I have been thinking about it since yesterday. I just haven't gotten everything prepared for it yet. Other questions? Yeah.
So when we do, so whenever we have paper copies.
Yes.
After class period, we just put it underneath the store.
If it's not, I think it's really key class.
Yeah. So preparation homework, if you've read the syllabus, preparation homework is due at the beginning of class. So if I wanted to right now, I would take up your statistics assignments right now. I actually want you to have them with you as we talk about things today, so I'll take them up at the end of class. Practice homework is generally due at 5 o'clock on the day that it's due, because there's no real need to have a deadline at the beginning of class because it's actually just practicing something that we've already gone over. And once again, that's all laid out in the syllabus in the section on homework. Somebody else, yeah.
Are you just gonna post our groups like on Moodle? Or?
Yeah, when I send you an email, it'll have the group breakdowns in there. Yep. We actually put you in groups on the basis of who's in athletes, Oxbridge majors, et cetera, et cetera. It's not a random assignment of people to groups. We don't have a lot of information to go on, but we have a little bit of information to go on. What else? All right. How did you guys find the statistics homework? Okay, it took so long. Some of you say it was confusing. What was confusing about it? Oh, the math at the end?
Yeah.
Okay. What about the stuff leading up to the math?
The coding in the art studio.
Uh-huh. What?
It was a train wreck.
Oh, it was a train wreck.
Yeah.
Did you come and see me?
I looked at the video and everything, and then, Ben, he's an RA, he's a biology major.
Ben Zahn?
Yeah.
Yeah. Mm-hmm.
He helped me, figure it out.
Oh, nice.
Yeah.
Okay, good.
But I figured it out, though.
Good. Awesome. Others? Confusing, long? Confusing.
Easy once you figured it out. Yeah, once figuring it out was hard.
Figuring out what part of it.
Like, I couldn't get, I just was doing it, I wasn't setting it up correctly, so I couldn't get the code to run on my computer.
Okay, so that was an installation issue?
Yeah, so
Okay.
I was just because I'd installed it, but you had to, activate the, our studio, the thing you download.
Gotcha.
first,
Oh,
you
yeah.
Activate it, and I, just didn't know, so I had
You loaded RStudio without BaseR.
I downloaded BaseR, but I had to, activate it on my computer.
Okay,
Before I could
alright.
get it to run.
Oh, okay.
So, well, I
Do you have a Windows machine?
What?
Do you-
I don't know.
Oh, really?
Yeah.
Oh, you didn't open the installer?
Yeah.
Oh, gotcha. Okay,
So I was
yeah.
just, lost.
All right. But you figured it out. What's that?
I was figuring out, to read the code up above, then it
Okay.
makes sense.
All right, good. Okay. So this is your first introduction to R. At this point, you don't have to know why it's doing what it's doing, because it's just more time to teach you how to do that than we really want to spend on it. Just trust that we've written a set of code that works and does what we want it to do. Did you, in the end, get the point of that first part before you got to the equations? What is the point? Pre-medis, what's the point of that exercise? That long, complicated, we're having to use this computer program that we're unfamiliar with sort of task. You're not a pre-medi. I'll come to you if they can't do it. Are we simulating a population, or are we simulating something else?
Simulating probabilities.
Sort of.
Possible outcomes.
Possible outcomes of doing what?
Drawing cards.
Of drawing cards from a population of cards. I'll give you guys a chance. Just don't worry, I'll come back to you. Yeah, you want to add something?
Me?
Yeah. What's your name again?
Katie?
Katie.
How different sample sizes affect like the correctness of the population, I guess.
So the different sample sizes affect the
accuracy of the
whether you reach the true population mean of whatever population is that you're sampling. In the back of the green shirt, remind me of your name?
Sonia.
Sonia.
Like the larger when you did like 1,000, and then it was much closer to the accepted value of 7 every time.
Okay, good. That's getting at what we wanted you to get out of that. Was it apparent that that's where we were heading when you started? No. Did there come a point where you're like, oh, this is what's going on? Okay. Sometimes we'll give you assignments that are like that. You will start down a path. You won't necessarily know where we're heading, know where we're taking you, but over the course of the exercise, it becomes obvious to you where the point is, what the point of doing this thing is. And we could just tell you about the relationship between sample size and the match of a sample mean to the population mean, but us just telling you that wouldn't drive home that point to you as well, and you wouldn't remember it as well than if you discover that for yourself. So what we did was we created an exercise that would allow you to discover that for yourself, and that should, in theory, have more resonance with you, and you'll remember that lesson better than if we just got up here and lectured about it. So that's the reason for that. And sometimes it just takes a little while and a little bit of work to get there. So trust us on these things. So this is about basic statistics, but I want to just go back and recap a couple of things from the COOT example. We talked about the scientific method, this process of making observations, generating questions, forming hypotheses. One way of thinking about a hypothesis is just an answer to a particular question. In the case of the COOTs, what was their broadest big picture question? Yeah. Why do the baby coots have bright colors? That's a better question than do the baby coots have this coloration because of parental favoritism? Because then the answer to that question is either yes it is or no it isn't. But this general question of why are they colored this way, it allows you to entertain a lot of different hypotheses, potential answers to that question. And we put those up on the board and we evaluated each of those for figuring out kind of which ones would be best to pursue. So just think of a hypothesis as an answer to a question. If the answer to the question is either yes or no, chances are you've already narrowed that question down too far. Then you design an experiment. We talked about how they designed their experiment. You have two types of experiments we talked about. Mensurative, where you look at natural variation to set up your experimental treatments. And then manipulative experiments where you, the researcher, actually alter the variable that you're interested in. Then you do the experiment, you get the data and you analyze it, and you come to some conclusion. And inevitably, the conclusions that you reach, what you find, generates more questions, and so you go back to the question generation phase, and you're just in this iteration of the scientific method. Starting next week, you will go through these steps over the course of the entire semester in lab. And you will present what you find in lab to the rest of your classmates in your lab, and you'll share with us what you did and what you found.
Maybe this is obvious, but we want you guys to be comfortable with the fact that when you think about hypothesis as a potential answer to a question, like why are these bright colors, it's fine if that answer is right or wrong. That's the purpose of the experiment. So just because you have a hypothesis, it's nice when the experiment and the data analysis supports that hypothesis, but it's fine if it doesn't, because that's why it leads back to.
Yeah, in the case of the coots, you could have examined the hypothesis that these are sexual displays. Now, you would have gone and looked for evidence to support that hypothesis, and you wouldn't have found any because, for one thing, they're baby coots. They're not having sex yet. So yeah, it's fine. Now, the goal would be to have a set of hypotheses that are good, viable hypotheses that make logical sense, and then you kind of choose from among those what you think your best bet is for being right. But you oftentimes guess wrong. And then you go test the other hypotheses when you find that the first one wasn't borne out by the data. So we're going to have you conduct observations. Another one of the homeworks from lab yesterday is to make a set of 10 observations as you walk around campus. And when you think about observations as being kind of trivial observations versus non-trivial observations. Trivial observations are observations that don't lead you down a path to discovery. The sky is blue. Well, that's an observation, but it's not really useful for anything. It's really hot today. Another observation that isn't particularly useful. The sky is blue, the water's cool, the flower is yellow. This is kind of biological, but okay, so what's the point? The flower is yellow. It doesn't get you anything. Non-trivial observations get you down the path to thinking about questions that are interesting. So these yellow flowers are covered by these yellow beetles with the black spots on their backs. So that is an observation that might make you think, okay, these flowers have these beetles. The flowers are yellow, the beetles are yellow. What are some questions that might come out of that simple observation? Yeah.
Why do the beetles match the color of the flower?
Why do the beetles match the color of the flower? Do they actually match it, or is it slightly different hues? Potential answers to that question is they might be colored yellow because they feed on the yellow flowers and they've evolved camouflage over time, et cetera, et cetera. So you can see why some types of observations are more useful for generating questions than other types of observations. Give yourself a point for that. That's my pen, so toss it back to me. Oh, you brought your own Sharpie. Oh, you came prepared. Awesome.
That was the
Okay. So we can also divide questions into mechanistic versus evolutionary. Another way you see this in the literature is proximate versus ultimate. Mechanistic deals with kind of how things happen, whereas evolutionary tends to deal with why things happen. So, for example, there are these moths called hawk moths. They look like this. Some of them have really long mouth parts. Some of them have really short mouth parts. And so one set of questions might be, how does a hawk moth extract nectar from a primrose plant? Why does a hawk moth have a proboscis that is so much longer than is necessary, given the length of the nectary? This is a mechanistic question. It's asking about the mechanics of nectar extraction. This is an evolutionary question that basically poses the question, why a proboscis this long versus a proboscis this long? What evolutionarily has driven one species of hawk moth to have this huge long proboscis, whereas other species of hawk moths don't have those huge long probosces? And so the first of these questions is more of a mechanistic question. Actually, I don't know why I have that under evolutionary. This is more of a mechanistic question. This is more of an evolutionary question. So I'll try and fix that on the PowerPoints. So this is something to keep in mind as you go into lab next week. Your question that you are thinking about as you go through the next couple of weeks of lab should be based in the observations that you make out in the field. Don't come to the lab experience with some predetermined notion of what you're going to see and what you're going to do a project on. Make good observations. Those good observations will lead to good questions, and that will lead you to a good project. They should be grounded in what's already known about the biology of that system. Yesterday's activity is to get you grounded in the biology of organisms so that you can come to observations with that knowledge in your head. They should be linked to some underlying conceptual framework, and many of these conceptual frameworks are in that list of 7 things that organisms have to do to survive. You have to acquire energy. You have to maintain water balance. You have to avoid becoming prey for something else. So you have to avoid being killed and eaten, et cetera, et cetera. All of those are these big conceptual silos within ecology and evolution. They should lead to a series of alternative hypotheses, and those hypotheses should be mutually exclusive. We had some mutually exclusive hypotheses on the board for the coots. Some of you said that maybe that serves as some form of camouflage, and then others of you said that maybe that's a way in which parents can find their babies. Well, those are obviously 2 mutually exclusive hypotheses because one depends upon being hidden, while the other one depends upon being obvious, standing out from the environment. And then the other thing that a question should do is it should point you toward a set of experiments and observations that allow you to answer that question. These are the characteristics of a good quality question.
Just I had a question in last lab that might we don't necessarily expect you guys to know the background biology necessary to make a really good question out of a good observation. And we're going to have time where if you identify good observations, then you can not only will you be able to think about and refine those, but you can also read about the organisms. And that's part of the process of developing hypotheses from good observations.
Yeah, so you'll make observations next week. Then I think for 2 more weeks, we spend all of lab in question development. And you think, oh my gosh, we're going to spend 2 whole labs, 2 whole weeks on question development? And the reason that we do this is because it's the question that drives us. If you have a crap question, you're going to, by definition, have a crap project. And so we take the time in lab, an inordinate amount of time in lab, to help you develop a question. We start with a bunch of questions. Each of you is going to present 10 questions. From that 10 questions of your group, so there are 5 of you per group, that's 50 questions that you start with. You're going to whittle that down to 5 questions that you think are good and worth pursuing, and then the following week we're going to whittle that down to 1 question out of those 50 questions that you began with. And so we're going to spend the time to help you with, well, how do you go about developing a good question in biology? Because we don't expect you to know how to do it at this point. So, questions about questions, I guess. All right. Ultimately, you're going to have to learn statistics. In this class, we are going to teach you some things about statistics. But the goal of this is not for this to be a statistics class. The goal of this is for you to have some fundamental concepts about statistics, and then we're going to teach you specifically about the types of methods that people are using in the papers that you're reading to do statistics. So we're going to teach you this thing fairly soon, right after the first test, called principal component analysis. I encountered principal component analysis in a multivariate stats class when I was in grad school. We are not going to teach you how to run a principal component analysis. That's ridiculous. For one thing, they're complicated. It's really graduate level work. But what you need to know is you need to understand what a principal component analysis is doing in a conceptual way such that when they talk about it in the paper, you're not just totally thrown by what the heck that is. And so I teach you principal component analysis with a three-dimensional model of a three-dimensional data set that you can pick up in your hands and you can turn it in different ways. Because essentially that's what a principal component analysis is doing, is it's taking multi-dimensional data and it's turning it in ways that maximize variation along new axes. And so we're going to teach you that intuitive idea using a physical model in class and also some real data so you can turn real data on a graph within R. But we're never going to teach you how to actually run it because that is way beyond the scope of this class. Same thing with analysis of variance. We're going to teach you actually how to calculate analysis of variance because it's so commonly used in science. But the main purpose of us doing that is so that you understand the analysis of variance that is in the paper for the second paper that you will read. And we'll teach you regression. Once again, another super common statistical method that is in the last paper that you'll read at the end of the semester.
One of those objectives on the first page of the syllabus is to be able to capacity to read and correctly interpret graphs. So having a sense of how to do these things, even if it's not at the level of doing yourself, you can read the graphs and look at them.
Yeah. So learning goals for today. There are two things that are crucially important for you to understand any statistical method. The first of these is the central limit theorem, which is what you were working on for homework. And the second of these is the empirical rule, which you also learned about doing that assignment, even though you may not have known that you were learning it. And these are two things that are crucially important to understanding how information gained from samples relates to other samples or the population as a whole. The empirical rule relates to 95% confidence intervals and why we use 95% confidence intervals. How many of you have ever heard of the empirical rule? Oh, nice. Nice. I know who's getting some points later. Understand why 95% confidence intervals are preferable to other measures of dispersion. Understand how sample size affects the width of the 95% confidence interval and one's ability to make inferences. And then you've already practiced what it means to estimate what a 95% confidence interval is for a data set. That was the end of the assignment with the complicated equations that some of you struggled with. And one thing, it's in small print here because it's a subheading, but this is actually crucial important. When you're interpreting two means and want to know if two means are different, it isn't about the overlap of the confidence intervals, it's about the inclusion of means in confidence intervals, and you'll see what I mean by that later on. But this is actually probably the most important piece on this entire slide, this little thing here at the end. So we have to deal with variation. It's natural in natural systems. You all look different from one another. That's variation. You're different in eye color, hair color, height, weight. Variation is just a part of nature. It's not bad to have variation. It's just that it requires that we are able to deal with it effectively. It can be due to environment, genetics, or human errors, or a combination of all of these things. Many people view variation as a bad thing. We do not. It's just biology. Things are variable. Not all flowers are the same. Not all insects are the same. So how do we deal with variation? Well, we deal with it by quantifying it. And that's what the latter part of your homework assignment was about. So we think about error. You'll hear people in science talk about error. And there are different kinds of errors. Some errors are fine and other errors are not so good. Sampling error is the error that you come into contact with most often. And it comes from when you get an incorrect view of a population of observations because you're only sampling a few of those observations. So It's a deviation. One of the other ways of saying this is it's a deviation of the sample mean from the population mean due to the fact that you did not sample the entire population. So if I wanted to know the average height of college students in the United States, I could actually just sample you guys. And it would tell me something about the height of college students in the United States, but it wouldn't tell me everything about height in college students in the United States. Because how many of you are on the basketball team? How tall are you? Oh, well, you're on the short side, I guess. Is that short for a female basketball player? Oh, that's about average. Oh, okay. So it's about average for a I don't know anything about basketball. I'm a scientist. So we lack a lot of basketball players, so our sample, because it doesn't include that, just by randomness, would not be a very good estimate of college students because quite a few college students who play basketball. How many people on the basketball team? 15. So we have 1/15 of the basketball team. So sampling error is when you don't get a very good estimate about whatever it is you're trying to measure about a population because you've just sampled a little chunk of that population. And we're going to use this, this image that I'm about to show you later on probably for another purpose. But as I was thinking about how to talk about this today, this came to mind. What do you see here? You see keys, don't you? They're car keys, house keys. They're keys. They're keys of different colors, so there's variation among the keys. And so if you looked at this small smattering of a couple of hundred keys, you might think that this is an image of just keys. But you would have that impression because all you can see are keys, and you can see a limited number of keys. So you don't have a complete picture, and why can I not see my cursor? Hold on, I'm having technical difficulties. In my office, I have 2 screens, so I have to turn off mirroring. I should have done that before I came down here. Yeah, so you can still see keys. That's a lot of keys. You saw keys because you were looking at a little portion of the population of keys in this image. So you had an incomplete picture, and not a very good picture of what the population is like. This is a guy named Chris Jordan. He does this kind of work where he basically creates images that are composites of other images to make points about things. You said it was a lot of keys. It's 260,000 car keys, equal to the number of gallons of gasoline burned in motor vehicles in the U.S. every minute. So the reason that we do statistics is so that we can look at a few keys and see the whole picture. But the more keys we look at, the clearer the picture is of what we're trying to see. So if we want to know about coots and whether or not their coloration affects their survival, for example, we can't look just at one family of coots, because that one family may just be dysfunctional, and all the coots die because the parents are terrible at parenting. So what we have to do is we have to look at how many pairs of coots did they look at?
90.
They looked at 90 pairs of coots, because by looking at 90 pairs of coots, you get a clearer picture of what the relationship is between baby coot coloration and survival and feeding rate and all those things than if you just looked at one family of coots or 2 families of coots or 5 families of coots. Okay? Does that make sense to everyone? I feel like I need to pay Chris Jordan because he uses artwork all the time. So that's an artist called Chris Jordan. He has very interesting artwork that is surprisingly relevant to ecology and evolution, strangely enough. So we have sampling error. That's just a function of the fact that you don't have infinite amounts of time and infinite amounts of money. If you had infinite amounts of time and infinite amounts of money, you could maybe sample the entire population. And if you sample the entire population, you don't need statistics. You know what the answer is. So you don't have to invoke statistics to make decisions about things. Unless you make measurement error. And measurement error is something that is within your power to avoid. Make sure that the instruments that you use are calibrated correctly. Make sure that you don't walk around using your meter stick as a walking stick, slowly eroding the length of the stick so that the meter stick is no longer a meter, things like this. So measurement error comes from gathering data in ways that are inaccurate. You can also measure data at different levels of precision and different levels of accuracy, and you can also have methods that are repeatable. So one of the things that we oftentimes see in first-year student writing in literature summaries is you guys tend to use the word accuracy in ways that aren't really correct. So I want to talk a minute about the differences between precision, accuracy, and repeatability. Have any of you guys ever had this conversation with a teacher? What's the analogy that people usually use to explain these things? Yeah.
The target,
The bullseyes of what? Search for it on the internet. This is what you will get. This is the worst, okay, maybe not the worst analogy, but it's not a great analogy. Because here is what they would say is not very accurate and not very precise. But the fact of the matter is that you can't tell anything about precision from this figure. This says higher accuracy and, once again, very little precision. Once again, this has nothing to do with precision. Precision is not illustrated there. Accuracy is about, does a measurement that you make reflect the correct measurement of the thing that you're measuring? If you measure something that is a meter long, is your meter stick actually registering it as a meter long? Accuracy is about, does it give you the right answer? Are you guys familiar with the international standards? Somebody tell me about international standards, or the U.S. standards for that matter. The Institute of Standards and Technology in Colorado, they have a meter bar. It's made out of some exotic metal, but it's a bar, and it's a meter long, sits in a velvet case in a vault in Colorado Springs, and it's the U.S. standard meter. It's based on the international standard meter, which is in a velvet lined case made out of some exotic metal that sits in a vault in Paris. Yes, ma'am.
In chemistry, Price talked about there was a European standard for kilograms.
Oh, yeah. Mm-hmm. Yep. Lost weight over time. Yeah. So there's a bar that represents the meter. This is not a meter. I don't pretend to know exactly how long a meter is, but there's a bar, and it's a meter, and it doesn't have a mark on it. It's just a solid piece of polished metal. It's accurate to a meter's worth of precision. It's accurate because it's the standard meter. If you want to know if your meter stick is, and you had a good reason, I can't imagine what the reason would be, they might let you into the vault. You could put your meter stick up against the standard meter, and you would be able to know whether or not your meter stick was actually measuring a meter. They're not going to let you do that. But if you did, that's one way of testing the accuracy of your measurement device. But my meter stick is divided up into millimeters. So my meter stick that I buy for $3 at the Home Depot is more precise than the international meter because its smallest division of measurement is a meter. You can't accurately measure a half meter on that thing because there's no marking for a half meter on it. You can't measure a centimeter on it. You can't measure a decimeter or a millimeter on it because there's no markings for those things. So precision is about how finely divided the measurement scale is that your instrument will actually measure. Now, it may measure that thing 1.5 meters off, but you can measure that 1.5 meters of error, of inaccuracy, down to the millimeter. So accuracy and precision are very different things, and they're not reflected in this at all. And then repeatability is about when you measure the same thing over and over again, does it give you the same answer? Now, it may give you an answer that is not particularly precise. It may give you an answer that is not particularly accurate, but every time you measure it, it gives you the same answer. That's repeatability. So I grew tired of these examples. So this is just what I'm talking about. Low accuracy, low precision. All of these darts are the same size, which means they're all of exactly the same precision. This is low accuracy, and once again says nothing about precision because they're all the same size. This is higher accuracy, but once again says nothing about precision. To know about these things, really, we have to break out guns and human targets. This is a human target being shot up by like a .44 Magnum. And because you cannot control the .44 Magnum because it kicks, you are not very accurate if you're trying to hit the bullseye. You are not very repeatable because you're not firing in the same place every time. And you're also not very precise because you're shooting the target with a .44 Magnum. A huge slug that leaves big holes in the target. These holes are placed in the same spots, but what's different about them? Smaller caliber. Same level of accuracy, not very good. Same level of repeatability because they're not all in the same spot, but they're more precise because now you're shooting somebody with a .22. I am not advocating that you shoot anybody with a .44 or a .22. I'm just saying this is more precise. Accuracy, still the same, terrible shot. Not very repeatable, but even more precise than either of those two things. This is precise because you're using a small caliber weapon. The holes are really small because you can measure things down to smaller areas. Very repeatable, but highly inaccurate. Every time you shoot the gun, it lands in the same place with a really small bullet, but you're way off the bullseye. This is really accurate, really repeatable, and really precise. Because this is now a pellet gun. Those are very small holes because you have things measured down to a really fine division. Be careful how you use terms like accuracy, precision, and repeatability. They're all terms that we use in biology and science in general. They have very specific meanings, and you should be precise about how you use them so that you use them in accurate ways. See what I did there? All right. Sometimes you sacrifice precision for repeatability. This is a thing called a Daubenmeier frame. It's used for measuring plant communities. And when you're in a plant community, it's sometimes difficult to decide how many of a particular plant is in an area. Well, this Daubenmeier frame is divided into quarters and then a 5% area down here. So that you can put this Daubenmeier frame down on a plot of, like in a prairie or something, and you can basically kind of mentally, in your head, place plants, group them together in your mind within this frame, and say, do they all fit within this 5% area? In which case, they would be in this first cover class. Do they fit, are they more of them that can fit in here, but not more than can fit in this quarter? Then they would be in the second cover class, etc., etc. It doesn't give you a ton of precision, but what it does do is when you're out in the field, you can say, okay, this yellow flower Would those fit in this 5% grid? No, it wouldn't, but they would easily fit within this quarter part of the grid. So they're in that second cover class. Not terribly precise, but if I give 10 of you a Daubenmeier frame, you're going to come back with the same answer. You're going to place each of the species in the same categories, cover class categories. And the other thing that this gains for you is by sacrificing a little bit of precision, you get high repeatability, and you get a certain amount of accuracy as long as you're okay with the lack of precision, and you also gain a ton of speed. It's very quick to assess plant communities in this way. You can quickly and easily train people to do it, and each person that you train to do it will do it the same way. So sometimes you don't need a certain level of precision, and if you don't need it, then why spend a ton of money and time gaining that level of precision if less precision actually gets the job done? And you'll see us making these kinds of compromises when we talk to you about your project. I used a $13,000 scale on my doctoral dissertation work, only to gather all my data and discover that I could have done it with a much less precise instrument that would have been a fraction of the cost. Luckily, we already had one in the lab, but it also took a lot of time to weigh things because you had to kind of put something on the scale and let it settle before it would register the right weight, and then you had to take it off. It had an internal door that was operated by a mechanism, and it had an external door. It was just a pain to use, and as it turns out, in the end, I didn't need that level of precision. I wish I had known that when I started. Questions about those things? You guys might use a Daubenmeier frame, you might not. It just depends on what your question is. So, these two key ideas about statistics, about numbers, about populations, the central limit theorem and the empirical rule are really important. So I gave you a task, which was to get a deck of cards and draw two cards out, average what those are, and then make distributions of these data. And I did it on the computer just like you did it on the computer as well. So we started with physical cards, but then as we needed more and more draws, it just became easier to do that on a computer. And what you find is with just 2 cards and 10 samples, you get a distribution that kind of looks like it might be normal if we turn our heads and squint real hard, but it's not a normally distributed curve. It doesn't have this bell-shaped curve that we think of when we think of normal curves. And the variability is quite high. It goes all the way down to just over 2, to all the way up to about 11 here. So there's a wide dispersion of the data. And the reason for this is that you're only drawing 2 cards. So when you draw 2 cards, you could draw 2 aces, which would make you think that the average value for that card deck is 2. Or you could draw 2 kings, which would make you think that the average value for that card deck, the cards in that card deck, are 13, when the actual average is 7. And so when you have small sample sizes, you get this incomplete view of what the population is that you are interested in knowing something about. And so I asked you guys to define what a statistical population is, and I haven't looked at any of your papers. Who wants to give us the definition of a statistical population? Yeah. Okay, you got that from a source, didn't you? Well, but where'd you get it? Like the Wikipedia definition? Okay, so repeat it again. Okay, so if we were thinking about the coot example, would the coots be the statistical population, or would something else be the statistical population? I just went over a thing that I wrote in pen earlier. What is the statistical population in the coot example? Is it coot families? Is it coot babies? Hmm? Is it the broods of the coots? Or is it none of them? Hmm? In this case, the statistical populations that these researchers are interested in are yeah, so a better definition for what a statistical population is it's the sum total of all the observations that you want to make an inference about. They wanted to make inferences about differences in feeding rates between black coots and orange coots. They wanted to make inferences about differences in the growth rates between orange coots and black coots. They wanted to make inferences about differences in survivorship of these two groups of coots. Now, you couldn't do that without the coots themselves, but the coots themselves are not the statistical population. The statistical population are the observations that you're interested in. Now, do they only want coots in British Columbia? Is that the statistical population? Or are they interested in making inferences about all coots everywhere? One would hope that they wanted to make inferences about all coots everywhere because that's the most general thing that you could possibly say. Now, did they go and measure all coots everywhere? No, they measured 90 families of coots in British Columbia. That's a sample of this broader statistical population that they want to say something about, but specifically what they want to say things about are these observations of different colored coots. So I realize that that's the Wikipedia thing. The Wikipedia thing is problematic because whoever wrote that definition hasn't really thought about it, especially from a biological perspective. What is a biological population? Oh yeah, and by the way, you got a point for being not complete. So good. Cheers for you. Yeah, what's a biological population?
It's a group of species living together.
A group of a single species.
Right.
Yeah, so in the case of the coots in British Columbia, they were actually sampling a biological population that happened to be these coots that lived in those three marshes, those three wetlands that they sampled. And so that could be a biological population. Because they were three different wetlands, it actually might be three different biological populations. But keep in mind that a biological population might not be the same thing as a statistical population. And that's why I want to concentrate on what are the things that you're actually wanting to make inferences about. And in many cases, you're not interested in the general organism itself. You're actually interested in one aspect of that organism. And so that's the thing that you're making inferences about. It's integrally attached to an organism, but it isn't the organism per se. That makes sense? Okay, cool. So what's the statistical population in the card example? Nope. The mean is something that we estimate about the population. What's the population? It's a deck of cards. And then you might be saying to yourself, well, this is ridiculous, Dr. Kowinski. We don't need to sample a deck of cards to know what is in a deck of cards. This is a stupid exercise. But the exercise was to make you think about if you didn't know anything about cards and you were given two cards to choose, depending on the two cards that you happen to randomly choose from the deck of cards, you get a very different picture of what that card deck is like. If you pick two cards, you get a poor view of what the card deck is like. You pick 5 cards, you get a better view of what the card deck is like. You pick 20 cards, you get an even better view of what the card deck is like. So as you increase sample size and you calculate something like the mean is going to be much closer to the true mean than any individual card will be. And it turns out that the mean of a bunch of means is even closer to the right answer. And that's basically what the central limit theorem essentially says, is that as you get larger and larger samples, the sample mean drawn from that population will be closer and closer to the actual population mean. That's what the central limit theorem says. So it's better to have a large sample size than a small sample size. I told you the other day, sample size of 30 is much closer to infinity than it is to 0, because you know a lot about a population if you have 30 samples from that population. You know almost nothing about that population if you have a sample size of 2. So that's how sample size influences how the sample mean relates back to the population mean. But seldom are we interested in the population mean. I don't know, have you ever been interested in a population mean?
No, I don't think I never had a paper where that was what I was trying to get at.
I've never had a situation where I was interested in the population mean, but I've had tons of instances where I was interested in whether this mean was different from this other mean. And in order to do that, we have to rely on the empirical rule. And I'll explain what the empirical rule is here in a few minutes. So, take 2 cards out of the card deck. We don't really know very much about the population. We can take 2 cards out of the deck 50 times. We still don't know very much. And what the program did for you is it plotted the mean, it plotted 1 standard deviation to either side of the mean, and it plotted 2 standard deviations to either side of the mean. What do you notice about that range of standard deviations? It's very wide, isn't it? It's wide at this point. If we increase that to 5 cards, what happens to the size of that range of the standard deviations? It gets tighter. It shrinks in. The mean of all these means is still close to 7, and it was reasonably close to 7 before. But in this case, the next one is now 1,000 samples, but each sample is still only 2 observations. And what you see is that the mean doesn't match up with what we know as the population mean, and the variation is still really big. But that variation is still really big because there's only 2 observations making up each of those means. It's still a small sample size. We've just done that same small sample size a bunch of times, so that rather than doing it 10 times, we've now done it 1,000 times. So we have a fuller distribution, and it looks like it's more approaching normality than it was before. 1,000 samples of 5 cards, 1,000 sample of 10 cards, 1,000 sample of 30 cards, 1,000 sample, 20 cards, and then 1,000 sample of 30 cards. What has happened to this distribution? The average of the means is closer and closer to 7, and the variation of this distribution is narrower and narrower. So the central limit theorem actually says 2 things. One thing that it says is that as you have bigger and bigger sample sizes, the sample means that you generate will be closer to the population means. But another thing that the central limit theorem says, and this is the best thing of all, is it says the following. Follow me carefully. This is one of the most important things I will say all semester. So remember what we did here is we took samples of 2 cards or 5 cards or 10 cards or 20 cards or 30 cards. We took those samples and then we averaged the observations within that sample. And then we made a distribution of those sample means, right? If you have a distribution of sample means, and if you plot this distribution of sample means, that distribution of sample means will be approximately normal. And it will be approximately normal more so if your samples are larger than if they are smaller. Now, why do I care about a distribution of sample means being normally distributed? I care about the distribution of sample means being normally distributed because oftentimes I'm asking myself, is one sample mean equal to another sample mean, or are those two sample means different from one another? And so I want to know about the properties of the distribution of sample means, not the distribution of the raw data themselves, because I'm wanting to make inferences about these sample means. Now, if I draw a sample from a population, and I get that sample mean belongs to a population of sample means out there in the world somewhere. But if I know that a distribution of sample means will be approximately normally distributed, I can use the empirical rule to now make decisions about things. Because what the empirical rule says is that about 67% of the sample means will be within one standard deviation of the mean. And about 95% of the population, of the sample means, will be within approximately 2 standard deviations of the sample mean. So if I have one sample mean and another sample mean, I can calculate how much variability there is around that sample mean, how much variability there is around that sample mean, and I can ask myself, what are the odds of me getting this sample mean from this population if it actually came from this other population? And I can do that because the empirical rule talks about how this relates back to the distribution of values underneath this normal curve. And so we use 95% confidence intervals because 1.96 standard errors of the mean includes 95% of the sample means. And so if a value lies outside of that, there's a less than 5% chance that it came from the same population that that sample mean came from. So it's this combination of the power of the central limit theorem and the rules about how normal distributions are shaped that allow us to say, is this mean different from this other mean? Or did they come from the same population, or did they come from two different populations, underlying populations? Does that make sense to you? If it doesn't make sense, stop me now because we have a couple of minutes that we can spend on this. Okay, think about it between now and Wednesday, because I have a couple more things to go through with you on this, and then Dr. Judd is going to take you through an exercise on Wednesday that he has homework for you for. I'm sorry, Friday, because today's Wednesday, isn't it?
Okay, you guys homework for Friday? I can, this little deal I know for you, I can send it out as an email message. But we're going to read a paper in class, like we did with the coops. It's going to be a different paper. We're here for that. We're going to use some stuff in class. These grades are stuff you use in class. So, in the folder for today, it's number 1. There's a text file. This is now TXT. Open that up. That is the text of the paper. So just as a text file, not as a PDF. So what we want you to do is to copy that and paste it into a word cloud generator. So there's a website called Word It Out. Word It Out. Just Google that. Comes right up. And create your own. You can paste the text into that. You'll get a word cloud. There's lots of word want to word it out. Don't read the table. You don't have to read the text at all. You do that in class.
Just cut and paste.
But your task is to try to infer what the paper is about based on the word count. Just based on the size, the relative size of the words.
Word clouds represent text where the biggest words that appear in the word cloud are the words that are mentioned most often in a body of text. And as they get smaller and smaller, they're less and less common.
So, just based on that, you're going to take out what the paper is about. But to help you develop that idea that that's what the paper is about, we also want you to make a concept map. So for those of you who were in the second lab yesterday, you have maybe a better idea of what a concept map is than those of you who were the first and you've never done it. There's a website called MindMUP, Mind Up, and there's another one called Bubble.us. These are both good ones. Here. Either one of those works. And as you think about the important ideas based on the words, organize those ideas as bubbles in your map, and then draw lines connecting them to what would the flow of the paper be? What's it talking about? How these different ideas that I'm inferring based on the word cloud relate to each other. It's an exercise in previewing the paper before you read it. Organize them into concise main points. That's what goes in your bubbles for your mind map. Use arrows to show relationships and connect those. Bring your best guess at what the paper is about in the form of mind map in class on Sunday.
All right, and hand in your homework to me. No, do not email it. We had this conversation earlier, didn't we?
I also
Thanks.
wanted to talk to you both. I can do it separately, but
Soccer, yeah.
I'm on the soccer team. My name's Clara.
Yeah.
By the way. And so I was gonna ask if you could sign this, and then I have a copy for you as well. I checked all the dates, and I won't miss any exams.
Okay, cool. Do you have a pen, or I might have a pen? You do have a pen. Okay, great. Oh, is this just to show that you gave it to me?
Yes, and I'll turn this copy into my.
Oh, come on. They don't trust you?
I guess not.
Let's see. 1 September 2021. Yeah. Yeah. All right. Thanks.
Thank you.
Yes, thank you.
Thank you. Thank you.
See, this is going to weigh down my satchel, and I'm going to get these back to you on Friday because I'm going to be sick of lugging this stuff around. I'm pretty sure I'm going to want to get rid of this pretty quickly. Okay. I'll keep them in order until I staple them together myself. Thanks. Yeah, that's another thing you guys should invest in is a stapler. I don't have a stapler. That's fine. I'll keep it in order, and then I'll find the boundaries.
So I wasn't really aware of how to do the cards.
Oh,
so
yeah.
I looked at the key and it looks like I cheated, but I more used it as a guide. So
I've been trying to delete the key so that people wouldn't look at it. That's fine.
I don't want to turn it in if it's
No, it's prep homework. Not a big deal.
Okay, sorry.
Thank you.
I have
Yes, sir.
class during your office hours. I know you want to meet those first 2 weeks.
Yeah, schedule time outside of officers. Just drop me an email with the time. Okay, thanks. Yeah.
What's your Thursday afternoon in terms of availability?
I'm relatively free. What do you need?
Awesome. I can
Two birds with one stone on the getting to know you.
Sure, we can do that. Yeah, I don't have anything this Thursday.
Afternoon wise tomorrow, I'm pretty much booked from 12 to 3.
3 okay?
I could do it at 3. I could do it before 12. You have lab though, right? In the morning?
So 3 works for me.
Okay.
3. Send us an appointment request, and then we will respond, and then it'll get on our calendars.
Gotcha. Okay.
Sometimes my signature that was at the bottom of the file, it evaporated. So
Oh.
I replaced it.
Did it go away when you got a new computer?
Yeah.
Yeah. It didn't evaporate. You didn't put it back on there.
I was setting in my account.
Oh yeah, it's a little file on your computer in Word. So I've got a little bit more to go through just the Finch data, so it's not going to take long at all to go through. So was the Chris Jordan useful or not?
I think so. At least I think the Chris Jordan is good. As soon as come back, I think they realize everybody can use.
Ah, okay. So I was trying to get a student to get the idea, and while I was doing it, I came up with the idea of a pixel. So if I had more time, and I might actually put this together, I think a better thing would be to do a picture that you have at different resolutions in terms of pixel size. And then as it becomes less and less pixelated, the picture becomes more and more clear. But I didn't have time to pixelate a picture. See ya.